or for the results repertoires that are derived from computational means, we might go after a more direct approach where we not only select and optimize the best binders, but we also think about the epitope surface, and we try to diversify the geometries by which we approach the target protein, and therefore hopefully affect the cellular activity downstream. So there's a lot of different ways to think about this. We can think about optimization, we can think about direct design. We could also take it in a different way and split it out by, do we have a starting material or do we have to go de novo? And so we'll come back to the different ways we can subdivide the problem later. Again, since I think here we have a pretty biologically fluent group, but let's just go through a little bit what the challenges are. So first of all, we're going to focus on antibodies for part of this talk, and the reason is just that antibodies are the composition of the starting composition of matter, so to speak, the fold that is sort of scaffolding, something like 80%, 75% of new drugs that are approved in a given year, that are truly new drugs. And again, this is because an antibody is a framework where we can use the immunization, the adaptive immune system of mammals to create new molecules with very high affinity. But the other reason is just that they have great half-life in the blood, and they have these C domains hanging off the bottom, which have lots and lots of other functions attached to them. They stimulate the immune system, they can affect cell killing. And so if you've never studied the functions that hang off the bottom of an antibody, it's actually worthwhile. It's a lot of fun. There's a lot packed into these constant domains. The part that we care most about typically are the loops in the heavy and light chain, the two domains here, the pink and the red domain respectively, that confer stability. So typically these L1, 2, 3, H1, 2, 3, these are the loops which approach a target and mediate very high affinity. And so if we look at this molecule, one might think, okay, this is a single fold. It's actually the same fold over and over again for the different domains. The heavy and the light chain are both what we just call immunoglobulin folds. But it turns out that there's a lot of function packed in here. Every region, every part of this antibody is doing something different. And then, of course, there are many different ways we can attach Fabs or single domain variants of Fabs or homologs of Fabs to different functions. So there's many different formats, but even if we stick with the traditional IgG format, we have a number of things that we might need to do. So these are large proteins, large multi-domain proteins. So keeping them in a solution that has the right properties with respect to viscosity, solubility, aggregation can be challenging, more challenging than many other things. The binding interface is mediated by strands and loops, which in computational structure biology are harder than helix-dominated interfaces typically. Or they have been traditionally. And then we have many things that limit what we can change. If we change too many things to optimize stability or affinity, we also might create sequences which are then cleaved and presented in a way that stimulates the immune system and causes the drug to essentially be rejected by the patient. So there's a lot of things that we really have to focus on simultaneously. And for us, that's exciting because it brings biophysics and multi-property optimization and experimental design and many other things that are essentially different subfields of machine learning together in a single project. The other thing to point out is just to zoom in a teeny bit to the CDRH3. This is what is typically the longer loop of these 6 loops that confer affinity in a typical antibody. The H3 loop, through the recombination and maturation through both chewback and mutagenesis, sort of multiple processes, the result is that the H3 CDRH3 can actually be quite variable in length. And what we find is that this is also a place where, because of the diversity that happens post germline, we actually see many different methods for predicting antibody structure actually do quite a bit worse on the one place in the antibody that we wish they did the best. Now, part of this is also because this is a region that is structured, in its functional form upon binding. That's by definition. And so to what degree is this because it's a long loop, and to what degree is it because there is likely some interaction with the target that also has to be considered to think through or model the functional form of this loop. Either way, it's one of the most important regions on one of the most important folds in biotech, and it's also one of the hardest to think about. And here we're looking at DBAB, A-body builder, IG fold, AlphaFold, and so we're looking at work from Google, but we're also looking at work from or methods from the Gray Lab at Johns Hopkins and from Charlotte Dean's lab, two places that have produced some really nice ways of modeling the structure of these loops. All right. Again, I've mentioned that part of why we're interested in antibodies is because they have just function packed into every atom. But another reason is that we have workflows that have been really super well developed in biotech over the last 20 years. Many times we start with the immunization of an animal. Could be a rabbit, could be a rodent. Sometimes we start with human antibodies. That's very, very rare. And there, of course, we're not actively immunizing a human, but we're pulling things from the population. And sometimes we look at antibodies from sharks and llamas and other strange places like that. And that's that's a bit rarer. Once we get those, next generation sequencing and many other lab techniques allow us to really dig pretty deeply into the pretty diverse repertoires that we typically get from high concentration immunizations. And that allows us to characterize those antibodies. But we still often have problems where we need to identify binders that bind in different places. When you immunize, when you use this sort of, let's call it this sort of standard workflow for discovering antibodies in animal repertoires, you don't necessarily get to choose where the antibody binds. In fact, you don't. You have to assume or hope that you'll get diverse binding modalities or geometries, but you very often run into cases where you need to repeat or optimize the immunization to get something with a better function and a different geometry. The other thing is humans and animals are different, and so there's a great number of steps that have to be taken once we find these binders to make them well tolerated, and give them good half-lives in the bloodstream of humans. So we have to so-called humanize these things. And then often when we do this process, we don't find great binders. There's a lot of really challenging targets out there, membrane proteins that have disordered regions, proteins that have very specific demands on the epitopes that we need to bind. And so there's quite a bit of demand for improving how we target antibodies to specific regions, and also how we deal with cases where immunizations or repertoires are not, where they've either failed or they're not available. And this is sort of leading into kind of a visual for what I just said, which is that if we have an antibody structure, we want to be able to model bound and unbound, and we want to be able to perhaps dock or at least characterize what part of the target protein an antibody is binding. And that might be because that's the basis for a de novo design method. And that also, you can imagine that would be a very handy tool to have if you wanted to sort through clusters of antibodies from a repertoire and pick ones that had properly diversified downstream function and geometry. All right, and I'm going to try not to run out of time here, but again, we have a lot of observed antibody space. We have lots of things where we actually know what the antibody is supposed to bind. We also have huge databases of naive or just wild type repertoires where we don't know what the animal or the human patient or volunteer has seen in their life, but we see what the circulating B cells, memory B cells, naive B cells are, and we can look at repertoires that way. So we have a reasonably large set of antibodies with their targets, a smaller set of antibodies with their targets with structures, and then a much larger set of naive repertoires. Here we're looking at a histogram that stops in 2019. And I think for the OAS database, if we then fast forward to a little bit closer to current, this is from a paper, I believe from the Grief Lab, we can see that there are actually substantial number of new databases out there, and including again synthetic, naive, bound and unbound, et cetera, et cetera. So there's really quite a nice set of sequences for doing things in machine learning, whether you're doing them in a self-supervised way or in a way that requires some annotation or development of a supervised oracle. Either way, the data's coming in. Genentech is also a place where there's substantial internal data sets, and a lot of effort is aimed at exploring these repertoires and building data sets, which from our perspective are just dreams come true for machine learning. And so I just want to thank a few of our collaborators who are driving, each of these folks are driving pretty big efforts. So Julian is actually scaling up the synthesis, testing, design, characterization of the antibodies that we design at Prescient. He's been working very closely with us and our engineering team to get it so that every 3 weeks we get essentially a new batch of designs tested. And of course, reality interjects, and that 3 weeks can become 5 weeks occasionally, but he has done a great amount of work to scale the biology of how we test things. Ryan, we need to thank for all kinds of things, but he's a world-class expert in using high throughput biology, including things like yeast display libraries and other cell-based technologies to screen large numbers of potential binders. And then Isidro is the master of the repertoire. He has all kinds of different methods. This is from a paper of his here, where we can see that we can use clustering methods, we can use machine learning methods, phylogenetic methods, but there's a great number of bioinformatic techniques that he's pioneered to get the most out of repertoires. And this sets the stage for where we started approximately, I think at this point, 2 years ago. We've been with Genentech for approximately a year. And Prescient is essentially a machine learning shop attached to this vast experimental enterprise known as Genentech. We also have a lot of support from Genentech Informatics. And with that support, we are going after first taking lots of current projects which have binders, which have starting points, which have molecules in different states of development. And we are developing methods for multi-property optimization of these binders, whether we're optimizing affinity or whether we're optimizing the other 16 parameters that you have to think about to make a drug. Again, with Isidro and Arvind and Yan Wu and many others, we're taking advantage of a lot of the great biology that happens at Genentech, and that biology allows us to characterize function and try to relate that back to the geometry of binding and rationally engineer that with a hybrid machine learning structure approach or with pure machine learning approaches. And I'll get into some of those details. And then lastly, the sort of grand challenge for all of this, and something that actually relates to many of the talks we've seen over the last 2 weeks, is the push towards de novo design. And here the twist is, of course, that it has to be de novo design of something that's still going to pass the other the other large set of drug-like properties, including immunogenicity and some of the formulation properties. I'll go through a little bit how we're breaking that down, or quite a bit how we're breaking that down, but I also want to just quickly walk through the process by which we got to where we are with Prescient integrated into Genentech. We were a relatively new startup. We hired a few employees and began the process in earnest when we were actually quite early in our pipeline, acquired by Genentech. And we joined Aviv Regev's sort of new push to bring data science and machine learning to the forefront at Genentech. We were also welcomed by antibody engineering and Arvind Rajpal, who also was relatively new to Genentech and doing quite a bit to bring new technologies in. Pretty quickly, we got our first round of designs with our first prototypes adapted to antibody data sets internally. And since then, we've been turning iterations on this overall design process. We're now on we're approaching our 7th round of designs for diverse targets. And we have a number of different methods that we are developing simultaneously. And so I'll talk a little bit about some of the basics of going after function and for an antibody, function begins with stability and affinity. I'll talk a little bit about multi-property, and then hopefully I'll have some time to talk about our ways of building that into an active learning framework, which is very important given the scale of testing that's possible at Genentech. So this is a beautiful slide that Stephen Ra put together with help maybe from some others, but this kind of just lays out visually what our overall focuses are in terms of how do we think about breaking down the problem for our machine learning scientists. And so I think right now we have a lot going on that is really focused in this representation learning space, and a lot of that is really surrounding efforts to connect the generation of sequences from powerful NLP-like methods with some of the really powerful representations for structure and for representing interfaces and other functional properties that are actually best expressed or most easily expressed as structural properties or geometric properties. And there we have had a few talks at NeurIPS, but one of the ones that we were all quite excited about was given by Jay, and that was Equifold. That was about a week ago in the structure biology workshop, so I won't talk much about that. We've also done quite a bit of work to think through error estimation and active learning. And there we actually have quite a few challenges. One of them is just that projects move quite quickly in a place like Genentech. There's a lot of new biology coming in, informing what we're going after, what we're doing. And so we'll talk a little bit today about active learning and how we do that when there's a pretty fast moving landscape of methods, data, and overall drug discovery objectives. And I've actually been really pleasantly surprised by how adaptable and how general some of the methods that Jiwan and Natasha and Kyung-Young and Stephen have chosen for us to use as our framework. It's really been quite amazing. The last thing on this slide is our sort of approach for generative models. And here, of course, maybe this is the basis for all of this, the way in which we actually wiggle the sequence. And so maybe this should be at the top of the slide, but this is actually where it all started. Our first product at Prescient was our Manifold sampler conditioned on function, which is really that entire technology was really quite focused on finding the best ways to make variable length generation for proteins in general, not just antibodies. Again, we have this arrow here at the top of this slide that talks, sort of uses let's say the proper drug discovery terms for the parts of the pipeline. But as we move through this pipeline, we don't want to think of this as a linear process, right? We don't want to think of this as a process where we get some leads and then we promote them one at a time and we have massive multiplicative serial dilution. We want to think of this as a process whereby which every time we iterate an experimental and then subsequent active learning and model retraining iteration, every time we go through a loop in this so-called lab in the loop, we want to update many of the different things that we're doing. We want to be able to improve the models so that late and early stage properties are integrated into a single model. And to do that, we do actually have to think quite a bit about our data and software engineering and how we interact with the informatics team at what is essentially a very large institution. We also have to make sure that whatever we're using can tolerate the growth in the model and the data sets that support the model over perhaps quite a few number of iterations. And then, of course, we want to use all of this to eventually combine our knowledge across a great number of projects and different targets to come up with general affinity or de novo design efforts. So again, this all started with the Manifold sampler. Pretty soon after we started working with antibodies, we realized that there were a great number of projects where even though we had the ability to make vast numbers of changes and approach this problem in semi-de novo ways, that in many cases that wasn't necessary. In many cases, we had really wonderful starting places, starting sequences, and we needed a lot of machinery to actually limit and impose boundaries and constraints on the sampling. And so this is work by Dan Berenberg, and Vlad Gligorevich, and many others at Prescient, which was actually in last year's NeurIPS workshop. This then led to lots of other explorations of how we can, again, make a generative model that allows a more seamless, a more foundational incorporation of different considerations, scores, oracles, and that's led to the adaptation of a new and the development of a new manifold sampler that's more consistent with energy-based methods that we want to explore. And as we go forward, we're seeing a lot more potential for integrating structure and sequence generation into single steps. We've seen a lot of really exciting work at this conference in that area. And so as we move through our technical roadmap, we're seeing that our LMS sampler, or our Langevin manifold sampler, is actually growing to include lots of other representations that are structure-based. Equifold is something that we sort of built not with structure prediction in mind, but with protein design in mind. And then, of course, we have diffusion models and other models in play that can actually integrate sequence and structure generation in really beautiful ways. All of the things that we do are physics or structure compatible. But I think one thing that we can say is that after our first year of hard work, we can focus on training, scoring, and sampling. We can focus on training and scoring and sampling function or dealing with function. The core method for suggesting mutations is something that is now, I think, well in hand. The other thing that we have to think about, again, is this idea that when we're discovering, or we're setting out to discover drugs or medicines, we have to actually consider a great number of properties. Some of them are categorical, or presented to us anyways as categorical. Some of them are biophysical. Some of them have very clear relations to geometry or structure, such as stability or affinity. Others are the product of the interaction of the molecule with a complex system like the immune system, right? That's immunogenicity and clearance. And so one example of our oracle is our expression oracle. And so when we design proteins and make a good number of mutations, we're also doing that again under the influence of a great number of oracles. Some of them are related to stability and affinity, but others are related to clearance and immunogenicity and other things like that. So we're making a good number of mutations, and we need something to babysit that process and make sure that we do not break the fold. And again, typically our designs can go as high as 50 or more mutations. And again, we don't always need that many mutations, but the method sort of naturally does make quite a number of mutations when we're in multi-property mode. Andreas, Lucas, Natasha, Ed, and Jan have put together an expression oracle we call Expresso that combines graph neural networks with structural considerations, and this can be integrated directly into our generative workflow. But right now we're at a point where, again, we can rely on generation of sequences from our manifolds. We can also rely on essentially a high rate of expression that's comparable to the rates of expression you get from pulling things out of B-cell clones. So we can move things a lot with, as I showed with the last slide, and we can also make sure that they actually are the things that we want them to be in terms of expressing and producing stable multimeric proteins. And again, this is a little bit more challenging in antibodies because they are somewhat complex multimeric proteins. They're beta strand dominated folds, and so they fold a bit slower than other things. And so this is something where if you get the structure wrong, the protein is very likely to be cleared. So I think this is actually quite a nice oracle. And I won't go into the numerous other oracles we're developing. Some of them are based on data that is actually kind of central to the drug discovery process and a little bit harder to figure out how to talk about safely. Okay, so once we have these things, then we do actually have to think about how to set up our push for general or target-specific affinity methods that that essentially are the heart and soul of designing an antibody. And at Prescient, again, we are integrated into Genentech in such a way that we're involved in a very large number of drug projects. Pushes to hit human targets that span the brain, the circulating lymphocytes that span the tumor microenvironment, but also targets that have different levels of structural characterization, that have disordered regions, that have membrane protein regions. And so we actually have a pretty diverse set of objectives. And so as we approach affinity, our solution to that is to actually try a few different alternative methods. So one question, and I'll run through just a few of our questions that we hope to have substantially more insight into by next NeurIPS, let's say. But one question is, how special are antibodies? How special is any fold that you've fallen in love with, right? Whether it's an antibody or an ultra large family of enzymes that have interest across biotechnology, who knows, right? But with antibodies, we do know that we have this loop dominated, very well characterized mode of binding, the three loops on each domain. Well, should we try to transfer knowledge from across the many millions of interactions where we don't have structure and we don't even have subdivision of the sequence into binding and not binding regions? I think there's a subset of us at Prescient that think we can actually do quite a bit with the methods we have to actually use a purely sequence-based method to suggest quite a bit of active functional cassettes and mutations for de novo design. And that's being led by Dan Berenberg, Vlad Grigorovich, and a few other people. Kyung Young is getting involved in this as well, and this is all very exciting. Another thing that we can do is take advantage of quite a bit of functional characterization at Prescient and at Genentech that spans libraries, saturation mutagenesis, and all these sorts of things. We can use that to start thinking about sort of a two-way generation and scoring, where we're actively trying to design the antibody, but we're also perturbing the target sequence computationally to evaluate hypotheses about the binding site that we're generating. And again, this is not really specifying an architecture, but specifying an approach here on this slide. And then lastly, one of the sort of big question marks is that it seems like there are a lot of very powerful methods for just co-generating one sequence given a specified region on, say, a target protein. But is that the right way to do that? Is there an advantage, or is there any value in actually trying to very carefully articulate the pose or the docking of the antibody to the target protein? How important is actually having an explicit loop out to structure when we're trying to design these things? And here, with both equivariant explicit structure representation, but also with graph representations, we have a good number of folks working on those types of both representations and hybrid approaches as well. One thing that I think we often do in science is when we find that we have a very good method, we don't always go back and do the comparisons to the prior work, right? And so here I'm showing work from Simon Kilo, who is one of our key scientists, working with Andy Watkins and Francisca Seeger and many others in our structure group. Simon has led the charge in using our various generative methods to replace the proposal of mutations and the proposal of moves in the Rosetta framework. And so Rosetta is still I think the code that is used for the vast majority of protein design projects on the planet right now. Typically, Rosetta has a great number of ways of sampling structure, but when sampling sequence, it is typically Monte Carlo, a random selection of sequences. There's a lot the user can do to specify what elements of the sequence can change to what. So there's a huge amount of configurability. But what we experimented here was, what if instead of any other proposal distribution, we strictly drew the proposal distribution from the manifold sampler, and then used an antibody redesign protocol from the Gray lab as the oracle. So this is just an experiment in taking antibodies and redesigning them there's a number of different things that Simon's gone after. This was one early success where he was able to use this manifold sampler with Rosetta as the final test, or the guiding oracle. And here we were able to improve binding by about 100-fold and improve some other properties in this molecule. And again, this was starting, so that we can show you, this was starting with a public structure from the PDB, from the paper shown here. But this is something that Simon's actively doing in the context of a number of projects now. All right, so if we have this variety of methods, and like I said, we also have a pretty wide variety of needs and specifications and objectives from the actual biology and drug discovery partners that we have across Genentech and Roche. If we have that really diverse landscape, methods and requirements, how do we decide how to fill up a plate? And even more complex, how would we decide to choose the mutations, regions, and just overall design of a larger library, such as a yeast display library or something even crazier than that, right? So we end up with these powerful methods actually substantially increasing the need for explicit multi-property optimization and active learning. And so I think you can find poster-associated talks for these papers, but I'd just like to highlight two papers that are pretty recent from the group. One is Property DAG, which is a way of the Jiwan and Sam and Saeed and others were able to come up with a way of dealing with the fact that many of the properties we're trying to optimize have relationships that make it hard to test one property if another property doesn't have a set of values. So there's a bit of a hierarchy built into some of the drug-like properties, and I'll enumerate that a little bit below. We also have to think about what's the best way to balance exploring space and improving models with exploiting the model and actually hoping to get something that fits the bill and can be promoted to a candidate for the further processes. These two papers are both, I think, really nice. And actually, the interesting thing about these papers is that they're being actively used in our real design cycle. And so there's quite a bit of good development of ideas in these papers. But Jiwan and Natasha are also down in the trenches using them to sort through the products of many different methods for many different projects every 3 weeks, ideally. So here's kind of just some of a little teaser to hopefully get people, induce you to go and check the paper out a little bit more carefully. But here's the crux of the problem, and so that is that we do want to be able to do lots and lots of things to these sequences and be much more aggressive in our designs, and we eventually want to really start this process primarily with de novo design. Well, if we do that, we have to worry about expression and yield because we're making a great number of changes. If we do that, well, if a protein doesn't express, how are we going to test if it does anything else? And the answer is you can't. And so all of our negatives, all of our zeros on the expression yield side can't be promoted to any other testing. And so then, of course, once you get a binder, once you get an expression, you can then test to see if the protein binds and you can characterize its affinity. But if you don't find that antibody has affinity for the target, it's very unlikely that then you're going to test its relationship or any of its further properties like clearance, immunogenicity, viscosity, anything else like that. And so what we find is whether it's through a biophysical reality like the expression binding hierarchy, or whether it's just an experimental choice to not test things that aren't binders, that's part of the culture, or they're just the overall protocol in the lab. Either way, there are very strong hierarchies built into the way in which we have to organize our active learning when the properties have these like very strict sequential relationships. And so again, that's I think really nicely dealt with here, and I think it'll remain an active topic at Prescient, but it's also something that is a very much case of building the airplane while you fly it, and we are using these methods in each round as we speak. So Another thing that I think we really have come to appreciate more and more as we build this airplane mid-flight is that when we have a very tight timeline on our lab in the loop, if we're suggesting thousands of mutations every couple of weeks, then we also end up in a situation where there are multiple versions of the model, and there are new data sets coming in. For a given design, you might see expression and stability, and then you won't see binding for a few weeks. And then there are other assays that take different preps and more material, and so you see those parameters later still. And so as we build through all of this, we actually have to be very careful in how we coordinate with informatics, how we coordinate with antibody engineering, and how we coordinate our own internal approach to organizing data and models at Prescient. Henri Dwyer, who's our lead for engineering, has been organizing a really nice way of dealing with this that deals with what I think he considers one of the key problems, which is that the process has a lot of asynchronous elements. It has a lot of elements where the timing varies, where one round might piggyback, hop over another round, and so there's a lot to sort out there. But Henri's been doing a great job, and the growing engineering team and informatics team at Genentech. It's been really wonderful to watch us automate more and more of this process. At the end of the day, we do actually have to figure out just how far we can push this, right? If we're building this vehicle that we can drive at high speed to discover new drug modalities, new affinities, new activities, to what degree can we take our lessons from that and actually turn it into an overall guideline for drug discovery across different modalities or different drug types? And again, our lessons here are the same. This is sort of a repeat, but again, we need to have the right methods and architectures to be able to get stability and affinity, but we also really need to think about how to take later stage considerations, more expensive experiments with less data, but again, really key considerations, reasons why drugs might fail late. We have to figure out how to get those considerations into the earliest steps of the computational protocol. So we need to have all the various parts of this timeline actually available to the samplers in the very beginning stages of drug discovery, and that's a very tall order. But that is the hope, that we can have end-to-end differentiable pharmacology, and that we can have properties throughout the full life cycle of a typical drug discovery project integrated into a single model. And we're getting there. It's been a really fun year. One thing that we can also ask as we work through this, and again, everything I talked about today, I drew examples from our experience with large molecule drug discovery, which typically means VHHs and IgGs and Fabs. Stephen Ra, who we met this morning, has also been thinking quite a bit about how much we can transfer our overall organizational knowledge in terms of how we use ML to approach LMDD to the space of small molecule drug discovery, or SMDD. And I think one thing we found at Prescient is just going through with our computational chemistry colleagues at Genentech and our colleagues in structural biology at Genentech, when we start thinking through the parallels between what we've learned with our LMDD pipeline and what we might do with SMDD and what we're doing with SMDD, we actually see a surprising number of really strong parallels. And one of them that's very clear is that active learning frameworks can actually, we can share a lot of know-how across the endpoint, the overarching framework that selects and diversifies what we try and that keeps track of and iteratively optimizes models. I think that is a very, very strong connection. The other thing is a lot of the efforts to obtain potency and affinity eventually come down to molecular interfaces targeting human proteins. And so although the different drug modalities we typically think of to generate agonists and antagonists are actually quite different, and the ways in which we find small molecules and make them, the ways that we find large molecules and make them, they're very different. Well, the actual underlying basis for that potency and affinity is still quite similar in terms of needing to diversify the ways in which you approach target protein and the things that you need to optimize affinity. We also have a great number of parallels in terms of the multiple properties that have to be thought through. So with LMDD, we do actually have a great number of properties. We don't really have to worry too much about how to synthesize our large molecules. We do have to worry about that quite a bit with small molecules. But again, there's been a lot of really wonderful recent progress on how to put that together and actually integrate synthesizability into a number of other considerations in small molecule drug discovery. And so I think at Prescient, what we want to do is say, listen, even though the way you make these molecules in the lab has led to pretty substantially different cultures of LMDD and SMDD units at Big Pharma, there's actually quite a bit we can do to share know-how, at least on the computational side, and produce joint models or at least similar protocols across. And again, another beautiful slide by Stephen, who gives our graphic designers a run for their money. Again, we have, this is just the basic science breakdown way of showing that many of the things that we're focused on for LMDD have very tight parallels in the SMDD space. So again, we have to think very carefully about how we, the data structures and the representation learning, but we still have to think quite a bit about molecular structure. Potency and affinity is still key. Our active learning is going to have substantially different properties that are incorporated, but we'll still have many of the same demands and motivations, including hierarchies such as if you can't make it, you can't test it, and a number of other things that are actually parallels to that expression binding other hierarchy that I showed you earlier that Jiwon and Sam work through so nicely in their paper. And then generative models and how they interact with multi-property optimization. Again, we do think that there'll be quite a number of parallels, but there's just been some really tremendously exciting work in this area for organic chemistry and biomedical chemistry there. All right, so I'd like to just go back and thank the team again. The column here is not all of Prescient Design. These are folks that gave me slides, let's say, and co-founders. The antibody engineering team at Genentech has been absolutely wonderful to work with. I can't go through it in detail because there's just so many facets of interaction that we've had to work through, so many things that we've had to learn, and they've been with us the whole time, building methods in the laboratory, finding new ways to scale biology, and really just innovating at every level. We've also had a lot of support from structural biology team, the DevSci informatics team, across GRED, including a lot of support from John Marioni and Aviv Regev to build out this team in the first place. And then it's a tremendously large organization that spans Roche, Genentech, and many others. It's really been great to have the support of the informatics team. And I'd like to especially shout out Samantha James for really working with us to get these somewhat complex data streams connected to our lab in the loop. And I think I'll end there and take questions or give a few minutes back to the organizers.
Great. Thanks for the excellent talk, Rich. So if folks have any questions, then please surface them in the Q&A and I can read them out as is. Otherwise, if there are no immediate questions, since you're in the hot seat, Rich, I'll ask one, coming into this initiative and knowing what you do now, having gone through kind of multiple iterations of this lab in the loop framework, how has your thinking changed about kind of the interaction with the wet lab and how maybe even methodologically we're approaching these problems? Yeah,
well, for me, I think one big way in which my thinking has changed is just really appreciating just how powerful the medi scale is, right? Like, how much do we rely on display technologies and other ways of doing massive diversification or saturation mutagenesis, for me, seeing the active learning interact with the roughly 1,000 design tests at a time, that seemed actually nearly as powerful as some of the more high throughput methods, at least in our hands. And partly that's because when you do these very high throughput tests of affinity or another property, you have to start with a given sequence. And so I've really, I guess I'm not sure I'm fully answering your question, but it surprised me how effective the METI scale is. But it's also led to a lot of questions about how do you take, not a small scale, but say like 1,000 designs every 3 weeks, and how do you integrate that with these higher throughput methods that can take a given sequence you're interested in, hyper explore the local neighborhood around there. So I think I've been surprised at how well things have been working. Pleasantly surprised, partly due to the quality of antibody engineering at Genentech. But I've also, it's led to a lot of questions about how far can we push this active learning framework when we're actually integrating these deep explorations into certain neighborhoods with the more broad exploration that we get from directing single sequence tests at the sort of multiple plate level. So a lot of my open questions are around continuing to think about active learning and what the right way to think about them, this mix of methods.
Great. And so we have a couple of questions that came up in the chat, and actually the kind of thinking about the active learning, I think, goes very nicely with this first question, which was, have you considered using reinforcement learning for the design piece?
Well, I mean, so that's also a little bit of question for you, Stephen, but we definitely have considered it. I think right now the basis for the generation of mutations, the core generative model is still not expressly that kind of a model. But there's a lot, we just got through 2 weeks of NeurIPS, and so I'm sure there's going to be a lot of really exciting discussions in the lunchroom in the next couple of weeks. So we'll see. But as of yet, not so much.
Yeah, I think there are a lot of exciting developments in the kind of like off-policy learning area that I saw in NeurIPS. And so I'm sure there's a lot to discuss in the coming weeks. So Christian has a question about what experimental measurements do you think we can't measure yet? So either high throughput or in general, and are necessary to drive the in silico development of large molecules? Great question.
That's a great question. Well, so first of all, let's take a step back from antibodies and just think about drugs, T-cell receptors, cell-based therapies, peptides, the whole thing, right? A lot of times we can very, very quickly, efficiently, at scale measure, did it get there? Is it made in one piece, and does it bind? And typically it's a lot harder to, or you can usually either measure that pure biophysical thing, or you can measure the activity, the effect on cells, and we don't always have the same methods available across affinity and activity, right? And so I think being able to separate those so that when you're measuring affinity, we also know it's important to measure the underlying biophysical properties and separate out successes and failures so that we know, again, that hierarchy. If something doesn't have activity, I want to really know if it doesn't have activity, but it has a very high binding, a very nice binding, because those are cases where you're actually learning about the function of the target protein. And so we can make those measurements, but it's not always the case that it's easy to align them, or it's in the budget to collect both kinds of measurements and align them. So that would be one thing. I think another set of measurements that we're excited about are some of the efforts to scale what are typically late stage measurements. I know we're very excited about measurements of things like viscosity and other properties that typically require higher concentrations, and there's some really exciting new methods that might scale those. I think if it was the beginning of NeurIPS, I'd give you a better answer there. part of my brain is still kind of leaking out my left ear from all the machine learning I've learned in the last 2 weeks. But that's a good question. I think if I think about that, I'm going to figure out a third part to that answer, like sometime tonight, that I wish I'd said. But yeah, that's a great thing to think about. But those are two of the things that I think we're excited about right now.
Great. And I think one last question we have time for. Let's see. What do you see as the biggest challenges going forward? So data, for example, complex biology, the availability of compute, different algorithms.
Well, I think for us, one of the places where we're most focused, where we feel like we might get the most, well, first of all of those things are really important, right? We're seeing a lot of improvements in how we can model not multiple sequence alignments or outputs from language models, but single sequences, which means it's design compatible. There's a real, I think, a really nice move towards models that are actually properly design focused. And so we're going to see a lot of movement in the underlying machine learning architectures, methods, representations that we use to do this kind of work. So I do think that's not a challenge that scares me, but it's like a challenge that I'm excited about. I'm really looking forward to what we collectively figure out as a field this year. Data is always a challenge. We want to take all the affinity data on the entire planet from every company and every public effort, and we want to put it all into one massive model. We certainly can't do that, right? And so every time you solve a data problem, you find that there's some social, business, or academic, frontier that you could cross again to increase the data set more, right? There's just a lot of data out there that's in different places, and I think very few groups have been able to train models on all the data that's out there. And there's no easy solution. The reason why data is scattered, to the four corners of the planet is because these are complex experiments. These are different targets. These things are happening at different places for different reasons. And also, like I said earlier, there's actually a surprising number of differences in the specifications and requirements of some of these projects. So I think getting data together into uniform databases and places where we can train models and keep track of models, I think that's a really big challenge that we should all go after together. Yeah. And that's something that we focus on a lot with our informatics team at Genentech.
I definitely agree there. All right. We're at the top of the hour. So thank you again, Rich, for a wonderful talk. And I think I'll be handing it over to Rebecca to announce what's coming up next.